Anybody have anything to offer?
Good morning.
Hi.
Um, Chemistry, yeah, it is a little challenging.
Okay.
So I've been just trying to just read up and like just trying to grasp the everything with the what's it like with the protons and everything.
Yep.
So I've been, just continually going over it and just trying to listen,
Mhm.
to the things that you said, on the things that you've uploaded.
Yeah, and the whole idea with all that is you don't want to get too bogged down. And I know it's detailed, but believe me, it's not as detailed as if you were taking a semester chemistry course. But really getting so that you understand hydrogen bonding, covalent bonding, and ionic bonding.
Mhm.
Hydrogen bonding we have to understand because everything we do is in biology, water is the solvent in our body, and water basically is a solvent we encounter in our daily lives. So we need to have the understanding of the special properties of water because you're going to be dealing with it here in this class, and you will in microbiology as well. But the real money will be understanding biomolecules, which we'll go over today. And so that will use some of those concepts. We don't learn hydrogen bonding for hydrogen's sake or ionization for ionizing sake or understanding, acids and bases. We only understand them in terms of how we use them in the body. So I decided to go over biomolecules in class, and if we get like one chapter behind or so, we get one chapter behind. We'll just have to live with it, we might have to drop something off near the end of the semester, and I'll figure out what that'll be as we go along. I updated the syllabus because of this. They didn't have, somebody was going to prepare like the skin and tissue lab to do online, and they didn't. So I long ago prepared things for a membranes, membrane transports. So we're doing that today instead. We're going to substitute for a skin and tissues lab, a membrane transport lab. But that'll help you better understand things for this next test. So that's probably pretty good that we're doing that. All right, let me try to get, I have this problem with my Mac a little bit. When people join in late, it's hard for me to move around the little screen. Okay, so can you all see biomolecules?
Yes.
Yes.
Right. Okay, so we'll start with biomolecules. We're going to use the concepts, hopefully, that you're starting to grasp from the chemistry and move on into building up our large biomolecules. There's Charity. Charity, for a while today, I might switch over to having you host the computer for like 5 minutes and show people how to sign on and load things up to Blackboard. So we might do a little fake thing like that when you did your assignment and you turned it in through Blackboard, because you're the only one, so I'm assuming everybody else doesn't know how to do that.
Okay.
Okay. And
Okay.
I obviously can't sign on to my Blackboard and turn something into me. So we might swap over at the halftime or something like that and take a break showing people how to, we'll go through all of our formatting stuff again. Okay. All right. So basically the things you're going to want to get out of today is we're going to go over identifying the four major structures or four macromolecules of carbohydrates, lipids, proteins, and nucleic acids. And so we're going to build those. We'll gain an understanding of how to use those. The idea of using these will become more important in the second semester when you take, study digestion and nutrition. But we also have like a lot of structural proteins that we'll be talking about in our body with muscles and bones. Probably not as much on lipids and carbohydrates. Also, we'll go over, as I said, the nucleic acids, and you just pretty much want to pay attention to a little bit the differences in DNA and RNA. Basically, they're different in their sugar, and they're different in that DNA forms a double helix where RNA is usually a single-stranded molecule. And we'll need to talk a little bit about ATP and how reactions, when they take place, either use energy or release energy. And ATP has a role in that, usually supplying the energy for various reactions we need. And we will only very, very briefly discuss some abnormalities or deficiencies of biomolecules. Okay, so the term organic is something quite different when we talk about it in chemistry, as opposed to when you're talking about going to the grocery store or buying, you know, Boll and Branch is constantly advertising organic sheets now, I see. Organic in the common usage of the term sort of means things that are safer with no pesticides on it, maybe not genetically modified organisms. But organic in chemistry describes specifically compounds that are based around carbon, and carbon that is hydrated with hydrogen. Because as I told you, hydrogen is the smallest molecule, 1 proton, 1 electron, and it kind of fills in the gaps on molecules all around. So long chains of carbons saturated with hydrogens will be known as hydrocarbons, or basically carbohydrates, okay? Our carbohydrates, as we know them in a nutritional sense, are usually called sugars and starches. And we all know that those supply us energy, and in their oversupply, they make us fat. You want to fatten up a cow, you feed it corn, right? So if you want to fatten up yourself, you eat a donut, nice starchy sugar. All right, so carbohydrates are the main source of our cellular energy. We burn glucose. Our brain preferentially burns glucose as its fuel over all other things. Glucose or sugars, they tend to be water soluble. They contain carbon, hydrogen, and oxygen, and they contain it in a ratio of hydrogen to oxygen that's usually close 2 to 1. And we can usually even say 1 to 2 to 1 in the ratio of carbon to hydrogen to oxygen. So C1H2O1, and when we get to glucose, C6H12O6. You just multiply everything. The size and the classification of our carbohydrates is we start out with our basic simple sugars. So this is going to be a theme as we build carbohydrates, proteins, and lipids, is what is the base unit? And as we link that base unit together, we build the bigger molecule, okay? So our base unit, or monomer, as we like to call it, is the monosaccharide, just one single simple sugar, like glucose, C6H12O6. When we link them together, when we link two together, we build up a disaccharide, which would be sucrose, which would be our table sugar, is a linkage of a glucose to a fructose. Lactose, which is the milk sugar, the same as the milk sugar that's in breast milk too, is a disaccharide. And when we link many, many, many of our sugars together, we polymerize it. So we have our monomeric unit to form our polymer. And our polymer is a polysaccharide, and that's going to be all of our complex carbohydrates like starch, glycogen, cellulose. And here is an example of how we draw out our sugars. We can draw it in a simple stick form, and it's an open chain, but you can see that this little open chain will kind of curl back on each other, okay? And where this double bond is on the oxygen, it will kind of link into the chain right here and form a single bond by bonding back over to this carbon down here. And you end up with a ring structure, a 6 carbon, or a 6 atom, I should say, because oxygen is one of the atoms. 6 atom ring structure like this. And this still gets cumbersome to look at because you have all these hydrogens and oxygens hanging off of it. So sometimes we just sort of have shorthand notation where you know that at every corner that isn't specified, it's a carbon, okay? And it's usually hydrated to some degree. So here we have our monosaccharide of a glucose, Here we have our disaccharide of a glucose hooked to another glucose, it looks like. And then we have them hooked together in long branches, long chains, and sometimes those chains are branching out, okay? And then we have some of our space-filling models of our important organic molecules. And so you have that six atom ring right there as a carbohydrate. And we'll build up lipids, and you'll see these long tails of fatty acids and, you know, so forth. We'll go on to our proteins and how our proteins fold, and our nucleic acids of DNA with our famous double helix. And I'm not going to test you like in super detail on the chemistry of this, but you still have to get a feel for these molecules because we'll be discussing them. And here's just a little secret I'll tell you is, you know, we make you go through all these really hard classes and all this anatomy, and your brain is going to be bursting and you're pulling out your hair trying to study all this stuff. And really on your day-to-day basis functioning as a nurse, you aren't going to have to remember all the gross details of anatomy. You'll remember what you need to know. So when you start feeling like you're freaking out that it's all not staying in your brain, don't worry, it doesn't stay in anybody's brain, okay? Even me teaching this and having been through med school and stuff, I look over the slides before I come and talk to you to like remind myself like, oh yeah. So don't worry if you think, you know, you just can't be a nurse or something because some of this stuff doesn't just stick in your brain forever and ever and ever. It won't. It doesn't for anybody. If they tell you it does, they're lying. And people lie a lot, all right? Your colleagues, as you're going through school, will freak you out, and they'll lie a lot, and they will posture all the time that they know more than what they do. They are no different than you. We're all human beings. We all forget stuff, okay? All right, you can all do this. I have faith in every single one of you that you can do this, okay? All right. So here we see the formation of sucrose. We have our glucose linked to a fructose, okay? And they're going to form a bond at what is the COOH end right here, the carboxyl end of the sugar, and this alcohol end, just an OH group, the functional group. And what they do is they form this bond and they lose the water. So this is called a dehydration synthesis, okay? We remove water and form a new bond right there. And so our new synthesized disaccharide of sucrose looks like this with a bond with just the one oxygen remaining, okay? So dehydration synthesis, and that will be kind of a theme. We're going to have lots of dehydration synthesis. We're going to have a lot of, also this could be called condensation, okay? You condensed two molecules together and you removed water. Just like when you have sweetened condensed orange juice in a can in your freezer, the water is removed. And when you want to reconstitute it, you put water back into it, right? So when we want to reconstitute fructose and glucose, we put water back into it and we split it back down into fructose and glucose. So that'll be a theme. Dehydration synthesis condenses the two molecules into one. And when we want to split this molecule of sucrose back into glucose and fructose, we'll add water and we'll hydrolyze it. We use water to split the molecule, all right? All right, so our next list on our list of organic substances is lipids, okay? Lipids are also long chains of hydrocarbons, but they're going to have a lot less oxygen content in it. If you go back here and you look at this, the sugar, you'll see there's like 1, 2, 3, 4, 5, you know, 5 oxygens on just the one basic sugar. And on a lipid, you'll probably find maybe like 1 oxygen, maybe 2, all right? What do we know about lipids? Lipids are fatty, they're slippery, they're oily, okay? So not having all this oxygen content, it reduces the polarity. When we were talking about our hydrogen bonds and our covalent bonds could be either nonpolar or polar. When they're polar, they have some charges to it, right? Even if it's just partial charges. Well, our lipids aren't charged really at all. And you'll see as we look at them, I'll want you to look at them and see that they're just long chains of carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, non-polar. And so when we mix oil into water, the two don't mix. You can shake it up, you know, and what happens? All those oil droplets will suspend for a couple of seconds, quick enough for you to hopefully pour your salad dressing on your salad, but it'll go right back in the bottle to the oil floating on top of the water. The two don't mix. They don't mix because like dissolves like. Water is polar and it will dissolve polar things. Oils and fats are non-polar, okay? And because they're less dense, they float. If they were more dense, they would sink, okay? All right. So lipids have much lower oxygen content and they will provide twice as much energy as the carbohydrates. Your carbohydrates supply about 4 kilocalories of energy per gram, whereas your oils will supply about 9. But I'm just telling you that you don't need to memorize that. Don't put that in your head to be memorized, okay? You'll get that next semester in nutrition. Amongst our oils, we're going to have a few different types. We'll have triglycerides, phospholipids, and steroids. Triglycerides are the main form of fat storage in your body. And this you need to know, okay? You'll probably have some sort of test question at some point. If not this semester, because you're not really having tests this semester, you're gonna have problems next semester, I guarantee. You will get a question on digestion. Triglycerides are the main storage form of fat. So when you think of your fat cells expanding with lipids and you're looking down at your thighs and your butt, or you're looking at Kim Kardashian's butt or something on TV, it is triglyceride heaven, right? All the fat is stored in droplets as triglycerides, all right? Phospholipids we know because we've all studied the cell membrane, okay? And we all have memorized the term phospholipid bilayer. When you think plasma membrane, the next 2 words out of your mouth should be phospholipid bilayer always. And then we'll get on to steroids, okay? So we'll discuss those a little bit today. But some of you have probably taken steroids. You know, you had an asthma attack and you went to the emergency room and they gave you some prednisone to kind of slow down the inflammation of your airways, something like that, all right? Okay, so our lipids are very, very important for our cell membranes. One lipid or lipid derivative that all of our steroids are based on is cholesterol, and that will be stuck throughout our cell membranes, and it kind of confers that fluid quality on our cell membranes so that they aren't stiff, okay? All right, so our most abundant lipids are triglycerides. Those are the ones that we'll store as the fat in our bellies, our bums, our thighs, all the places that, you know, oh, we love our triglycerides, don't we? Okay, and again, they contain carbon, hydrogen, and oxygen, but much less oxygen. And so we'll see now we're going to form our triglycerides, which will consist of 1 molecule of glycerol and 3 fatty acids. Okay. Just another thing to remember is I put out this quickie slide to remember that they're water insoluble, they're critically important. Remember, the primary thing for our lipids is the energy source, although we have that structural role as the integral parts of cell membranes. And here's what a fatty acid looks like. Okay, so you have a carboxyl group at one end, and if you don't remember all your groups that you learned in Bio 155, this COOH is a carboxyl group. And when we draw it out, it looks like this: a carbon double bonded to an oxygen, and the alcohol here part at the end, okay, the COOH. And the R group just means everything else that's on the molecule that's going to vary molecule to molecule. And I drew it out over here also just so that in case you forget, at the little corners are all going to be carbons, all right? So you can see that this long R part is going to be relatively nonpolar. but you have this little polar end, right? Because you have these oxygens with these, with its 2 lone pairs of electrons on each of these oxygens. And this hydrogen is gonna fall off this molecule very easily. It's an acid, right? When hydrogen ionizes and goes into the water solution, it becomes an acid. So we'll learn that our fatty acids may be saturated with hydrogens, in which case we're going to have carbon, hydrogen, carbon, hydrogen, carbon, hydrogen as our group, or we'll have some unsaturated fatty acids. And that's where we throw in a couple of double bonds of a carbon double bonded to another carbon instead of being saturated or surrounded with hydrogens. We say it's monounsaturated if our fatty acid only has 1 double bond in it, and we say it's polyunsaturated if there's more than 1 double bond. And we'll look at these in a minute. Olive oil is our famous monounsaturated fatty acid that we consume, and is, like, you know, probably the best oil for us to be consuming. All right, so here's looking at our fatty acids and what I mean by saturated and unsaturated. If we look here at this group in A, you see all these long, long chains of carbons saturated by a hydrogen. Every carbon has 2 hydrogens on it, and it's hooked to 2 other carbons, forming this long chain, all the way down to the end where the last one has 3 hydrogens saturating it. And of course, it has that carboxylic acid on the other end of it. And that carboxylic acid is what obviously puts the acid to the fatty acid designation. Our saturated fatty acids, you'll see here, has 1 double bond right here, and that kind of throws a little kink in the molecule. And then we have another double bond right over here. So this will be a polyunsaturated fatty acid, more than 1 double bond. All right, and now we're going to hook our fatty acids onto a molecule of glycerol. So when we hook 3 fatty acids to 1 glycerol, we've formed our triglyceride, and this is how we store our fat in our body, okay? So you have 1 long unsaturated fatty acid here, you have another unsaturated fatty acid, and here you have a monounsaturated fatty acid. Saturated, saturated, monounsaturated. Yeah. Starts to not roll off my tongue. And here's the glycerol, and you'll see this ends up being called an ester linkage right here, that we esterify our fatty acids to our glycerol molecule. And of course, we take away water usually when we're making this, so it's another dehydration synthesis when we're hooking our fatty acids onto glycerol. And of course, when we want to split it off, we usually use water once again to hydrolyze it, okay? All right, here you can see a nice molecule or space filling model of a saturated fatty acid, and you see how those long tails hang off of the molecule of the glycerol, and this would be a palmitic saturated acid. Here you can see how these double bonds in linoleic acid throws in kinks into the fatty acid tails, okay? So kinks. All right, and as I said, when we're forming our triglycerides, the theme remains, we're going to remove water as each fatty acid is bonded. So we're removing 3 molecules of water. So again, it's a dehydration synthesis or a condensation. And then we form our triglyceride molecule. If we want to go back over here to the individual glycerol and the fatty acids, we'd reverse this reaction, and it would be called a hydrolysis reaction. Okay, other than our fatty acids and our triglycerides, when we have one end of a phospholipid, one end of our fatty acid becoming phosphorylated, it becomes more water soluble, okay? And the other end is fat soluble. And this is how we then end up with our phospholipids that make up the bilayers in our membranes. So let's show a picture here, because this is a lot easier to understand when you look at the picture. So here you have one fatty acid and another fatty acid, and then on the end you have this phosphate group, okay? So you can see at that end forms that hydrophilic head. And you can see this phosphate has lots of oxygen surrounding it. So with all those lone pairs of electrons and a double bond, and here you have also a nitrogen compound. So this is going to be intensely polar. And when it's polar, it likes to go and dissolve in water, and we call that hydrophilic, okay? So when you take your HESI exam, if you haven't taken it, roots and stems, hydro always means water, and then philic means loving. So something that's hydrophilic is water loving, okay? So it wants to face the water. All these long tails of hydrocarbons are hydrophobic, or hydro water and phobic water fearing. And so as you've probably learned in the past, I hope, your hydrophilic heads of your phospholipids, one side will face the outside of the cell, and the other side of the bilayer will face the inside of the cell, because on the inside of our cell, we have the watery substance of our cytosol, and on the outside of the cell, we have all the body fluids that the cells are bathed in. So we have our water-loving faces of our phospholipid bilayer facing the water, and then in the center of it, we have all of our long fatty acid tails oriented toward each other, and they exclude the water. So this is why water from the outside of the cell isn't always rushing into the cell, and why water from the inside of the cell isn't always going out, okay? So we can kind of compartmentalize everything that we need to compartmentalize in our cells. If we want to look at our phospholipid molecule up closely again, we have our phospholipid heads and our fatty acid tails. And of course, here's the space filling model. Here's that hydrophilic head. And you see out here, you have this nitrogen containing compound. You have this really polar phosphate group. Okay, and that phosphate group we'll see again when we get to ATP. And then you have the glycerol portion, and then you have the long fatty acid chains. They didn't draw the whole thing out here. You can see it's CH2 over and over again 12 times, all right? And this is how your phospholipid bilayer orients itself in the cell, facing the water on each side, right? Internal to the cell, external to the cell, again, external to the cell, and internal to the cell. Okay, another type of lipid is our steroids, okay? Now, don't sit around, just memorizing all these chemical compounds. What you need to do is look and see if you can recognize this kind of 4-ring structure like this without all this junk, sort of hooked onto it. And when you see this 4-ring structure, you're basically dealing with some steroid or derivative of steroids. Okay, so it's 4 connected rings of carbons, 3 6-carbon rings and 1 5-carbon ring. Okay, our steroids are going to have lots of different functions throughout our cells. Cholesterol, like I said, is always going to be a component of our cell membranes, along with occasionally some other cholesterol derivatives or steroid derivatives. And we're going to use cholesterol as our base molecule to synthesize all of our adrenal hormones and our sex hormones. So cholesterol, from cholesterol, we're going to form testosterone, and we're going to form estrogen. It is the main steroid in the body. Cholesterol gets a bad name. Years ago, everybody thought that if you ate too many eggs, because eggs are in high cholesterol, it will raise the cholesterol in your body. And that turns out not to be so true, because when we ingest our food, we digest it, we break it down, and it becomes all kinds of other things in our body. The main cholesterol, 75% of the cholesterol in your body is made by your own liver. And this is why our statin drugs work so well to control people who have high cholesterol issues. It basically damps down the ability of the liver to synthesize de novo, or make new cholesterol from the liver, okay? So lots of people have problems with high cholesterol levels, and it's usually not because of what your body is ingesting. It's usually because of what your own body is making. All right, again, here's cholesterol, and you can see how our other hormones are derived from it. You know, you chop off a whole bunch of stuff here, maybe throw in a double bond over here, but you can see that it's basically based on that ring structure of cholesterol. And that's all you need to recognize with it. Okay, so steroids. What's all the fuss about steroids? And also prostaglandins. You'll hear about prostaglandins. And prostaglandin sounds a lot like prostate gland, and that's true because our prostaglandins, when we first discovered them, they were discovered in the prostate gland, but they are hormonal type substances that are throughout your whole body, throughout all the tissues. All right, so our steroids are very important in biology, chemistry, and medicine. Well, when you get to reproduction in the second semester, you'll study all the sex hormones, and you'll also study the adrenal cortical hormones. And for digestion, you'll just also study the bile acids that are made by your liver and stored in your gallbladder, and they will help you emulsify all the fats that you eat. They basically form like a little detergent that allows the fats to move through your digestive tract. Our synthetic steroids are also of therapeutic value. Like I said, if ever you had an bad asthma reaction and you end up in the emergency room, or your child ends up in the emergency room with an asthma attack, you're going to get a nice injection of prednisone, and that prednisone takes down the inflammation. People also abuse anabolic or growth stimulating steroids, okay, to make themselves more muscular. And, you know, of course, a lot of us ladies, we would take oral contraceptives, which are a form of steroid, okay? They're estrogen analogs that you put in your body so that you can avoid pregnancy. Another picture just to remind us of some of the derivatives. I probably put too many of these in here, but just like I said, you'll always see the 3 6-carbon rings and the 1 5-carbon ring, cholesterol, cortisol, etc. Our prostaglandins. These prostaglandins are derived from a fatty acid called arachidonic acid, which is in the membrane of every cell in your body. And our prostaglandins are derived from 20 carbons, and they form 5 rings. And they have a variety of effects throughout the body. You just think of prostaglandins as tissue hormones that come out of the cell membrane itself, okay? Tissue hormones. That should be just like when you think plasma membrane, phospholipid bilayer, think prostaglandin, think tissue hormone would be the 2 words you connect with it. And you can see here how our prostaglandins are a little bit different because it forms kind of like a ring structure, but it's not always like a defined ring structure, okay? And they're released from our cell membranes. Okay, moving on from our lipids, we did carbohydrates. Our monomeric unit of our carbohydrates is our sugars or our monosaccharides, okay? Our monomeric units for our lipids are our fatty acids. And now we'll get to our proteins, which are made from a combination of the monomeric units called amino acids. Okay, proteins are the most abundant molecules in our body, and they're basically, they form the structural weight of our body. You know, when you have a muscle, it's made of protein, okay? Proteins are also made from carbon, hydrogen, and oxygen, but they also have a new one in there, nitrogen. So we think of them as being characterized by the presence of nitrogen, nitrogenous proteins, okay? And every amino acid will contain a amino end or a nitrogenous end of the molecule that you'll see. We have 21 basic amino acids. They're small water-soluble molecules, and 8 of them are essential. And when we say essential, it means it is essential that you intake them from the diet because they aren't made by your body. The other 13, to varying degrees, we can make ourselves in our body. Okay, so our proteins will form the structure of our cells, our tissues, our organs, and it's the unique shape of the proteins that will give it its functions. It will allow it to fit in with certain other chemicals. And so we talk a lot about the three-dimensional structures of our proteins, okay? Our proteins will first form a primary structure, which is just going to be the chain of what are the amino acids held together. So is it valine, leucine, glycine, histidine, and so forth. That's the primary structure. Then it folds on itself. When we want to hook our amino acids together, we're going to do what else? A dehydration synthesis, which could also be a condensation. And just to remind you, when we're dehydrating, we're going to remove water, but we're going to synthesize or create a larger macromolecule. And we'll create it between the amino group of one amino acid and the carboxyl group, our friend, COOH, from another amino acid. And the bond that forms is going to be called a peptide bond, okay? And the resulting molecule is a small protein called a peptide. And when it gets to be bigger, we call it a protein, okay? So peptide is just like mini protein and has a peptide bond in it. And we don't really call the others a lipid bond or a carbohydrate bond. So peptide bond is really the only one that you have to be concerned with having its specific bond name. Okay, so peptide bond formation. This is the dehydration synthesis, removing water again. You can write in here dehydration synthesis or condensation if you want to. And then you form your whole peptide right here. So one amino acid group, you take the COOH end, the carboxyl end of one, and you hook it right onto the amino group of the other. And you knit these two molecules together by removing water, forming the peptide bond. All right, right there, that blue bond right here is the peptide bond. Okay, so then with our proteins, after we hook our amino acids together, we get a primary structure, which is just the order of the amino acids that have been hooked together, methionine, valine, histidine, leucine in this case. And then these will kind of twist on each other and maybe form some intermolecular bonds, say like between the histidine and the methionine. They might have like a hydrogen bond or something. And you'll find they'll form some helical structures, things like that. That will be its secondary structures. And then even some of these secondary structures might fold again over on each other and make some tertiary or third degree structures. And then we'll have some bigger, fourth degree structures. These are both tertiary structures right here, a beta pleated sheet. And this would be like a leucine zipper. Okay, but you don't need to know that. Our amino acids, again, we have our functional groups on our amino acids. So we have an amino end. So that looks like amino or ammonia, which is a nitrogen with 3 hydrogens surrounding it, okay? And because this is very polar, just like with water, the nitrogen pulls the electrons toward it and all of our hydrogens get exposed, and especially one usually will end up carrying a very positive charge on it. Then we have what's called the alpha carbon or the first carbon, and that's the one that's attached to the nitrogen. It will have the R group, all the other stuff that comes off of it that makes it its unique amino acid. And then it will have the carboxyl group at the other end also attached to the alpha carbon, okay? You don't need to remember alpha carbon. You just need to be able to identify that an amino acid has an amino group and a carboxyl group. And the R group is always whatever else is different on it. Okay, busy slide that makes your head want to explode. I know it does mine, so I'm sure it does yours even worse. Again, we have a dehydration synthesis here that's showing you a serine, a valine below it, a tyrosine, and a cysteine. And you see we're removing a water, removing a water, removing a water in each case and forming a new peptide bond that's between that alpha carbon of the carboxyl group and the nitrogen, okay, of the amino group of the next one. So you can predict the order going on through all these. As I said, if we want to break up this polypeptide that we made back here, we'll do it by adding water back in, which is called hydrolysis. And we break the bonds and we go back to our original serine, valine, tyrosine, and cysteine. Okay, so reversible. So I hope you're getting the idea that we have dehydration, synthesis, or condensation, forming our bigger molecule. Hydrolysis is always going to break up our big molecule into smaller things. So when we get to nutrition and you start consuming that nice, big, red, rare, juicy protein steak that you had on Labor Day, right? Or your burger, and you flip it over and you put it in your mouth and it goes down into your gut and you're enjoying it so much. All this water and digestive juices with enzymes comes into that food, and it's going to start hydrolyzing all that protein from that cow and make it into all its component amino acids that you will then absorb in your gut and send it out to the other parts of your body, okay? And then actually, you know, and then you'll use all those component parts here and you'll resynthesize different proteins that your body needs to make your own muscle, right? You don't just consume cow muscle and it's just somehow, you know, becomes your muscle. You have to break it down and build it back up. All right, so our levels of protein structure, this is something you will get tested on. So know that the primary structure is the amino acid sequence. The secondary structure is when it pleats or twists and makes these unique little secondary structures that are kind of little localized structures. The tertiary structure is when it folds back those alpha helixes on itself. And the quaternary structure is when you take two or more polypeptide chains, and you'll see this when we get to hemoglobin, and we'll have one sort of polypeptide chain, then we'll have another one, and we'll put them always associated, the two together with each other, okay? I'll tell you right now, where people always have the hardest time figuring out is between secondary and tertiary. So for secondary, kind of memorize the pleating or the twisting. The helix or the pleating are secondary structures, and then you usually get it. Tertiary, it's always going to be about folding back on itself. All right. So the importance of the protein shape will come into play, particularly as we talk about enzymes. And our structural proteins will have very specific structures that will, you know, form the unique parts of the body. When we have collagen, and we need our collagen to be nice and silky yet strong, like to cover the tips of our bones and help attach our muscles to the bones and get that like glistening white shiny collagen that you see when you do have your steak or something, the gristle you call it. It has its own unique structure, okay? So proteins themselves, their structure can be denatured by changes in pH, changes in the temperature, radiation, other chemicals. When you take an egg, which is largely protein, except in that yolk you have a lot of cholesterol, but the whites especially are a protein. You flip it over onto your frying pan and you fry it, and you've just denatured that egg, right? You've raised the temperature and you've denatured it. You've made that egg white turn into something that it previously was not, and it's never going to go back to being the other form, okay? So you've completely denatured it. That's the example I have for denaturing a protein that we can all understand. All right. So proteins will have a whole bunch of different shapes. You'll love this shape when next semester you get into immunoglobulins, something we're all talking about a lot now with coronavirus. As I said, collagen, we have our long strips of collagen, and you can see it's uniquely shaped differently from the immunoglobulins. We have troponin, which our heart spills out in its muscle fibers, and we'll survey for that when people are having a heart attack. So, you know, obviously you don't need to memorize these shapes, but it's just showing you that proteins can come in long, thin glistening forms or highly globular form, that they're actually even given globulin in their name. So, all right. And like I said, you can take the protein that has its specific structure and you can either raise the pH or raise the temperature and you can denature that protein. In some cases, you might be able to return it back to its native state. In other cases, like frying an egg, you've denatured it so much, you ain't ever getting it back. Okay, now we move on to our nucleic acids. We all love DNA and RNA. So DNA, we know, carries our genetic code. RNA we'll use to direct the code in DNA to make our proteins. All right, so encoded in the amino acid sequences of the proteins, all right, is the recipe from the DNA, all right? DNA has the recipe. RNA will take that recipe from inside the nucleus out to our ribosomes and tell our ribosomes how to knit together all the amino acids to make our proteins. Our building blocks for DNA, we had amino acids are the monomers of proteins, fatty acids are the monomers for our lipids, monosaccharides are the monomers for our carbohydrates, and now our monomers for our nucleic acids are nucleotides. And our nucleotides consist of a phosphate group attached to a sugar, attached to a nitrogenous base. So 2 substances have nitrogen in it, and that will be our proteins and our nucleic acids, okay? All right, DNA, as I said, carries our genetic code, your own unique recipe for making you, and directing the construction of all your body parts that need to be maintained or for growth and development. And DNA will consist of the 4 basic nucleotide building blocks. We'll have 2 that are pyrimidines, those are thymine and cytosine, abbreviated T and C, and 2 are purines, adenine and guanine. So you got to get it through your mind always. T equals C, or T and C always are the, I'm sorry, it's A equals T, because you always bond a purine to a pyrimidine. But remember that thymine and cytosine are the pyrimidines. I know we're going through this fast. Is your head spinning yet? Probably. All right. Okay. DNA, the differences between DNA and RNA. DNA has a sugar called deoxyribose, where RNA will just have ribose, okay? DNA will form double helixes, where RNA typically will exist as a single chain. And when DNA binds to the molecule or the nitrogenous base, will share a bond with from one chain of DNA in the helix to the other chain in the DNA helix. You will bind one pyrimidine to a purine, and adenine always exclusively binds to thymine, and cytosine will always bind to guanine, okay? So A always coordinates with T, and C always coordinates with G. And so this is how we see it. We have our sugar and phosphate backbone running in one direction, where you see all the phosphates and all the points of the pentose of the sugar of deoxyribose pointing up. And over on the other side, our phosphate's going to point out, and the pentose point is going to point downward. And we see our hydrogen bonds, once again, another theme, our hydrogen bonds of our adenine is going to be bound to our thymine, and our guanines will be bound to cytosine, A and T, C and G. There's 3 hydrogen bonds between C and G, and 2 hydrogen bonds between A and T. And remember, hydrogen bonding, each bond itself is really not a bond. It's not covalent or ionic. It's kind of a loose association. They're attracted to each other because they have partial charges on those nitrogenous bases, okay? Which you see down here. You have those hydrogens hanging off, and that hydrogen has a positive little charge on it, and it's attracted to the somewhat negative charge on the oxygen over here, okay? So just like with things in water, or water molecules bonded to other water molecules, they're kind of a loose attraction to each other. They can break easily individually, but the sum total that you have millions and millions of these base pairs going down a strip of DNA, it holds it together, okay? The sum total interaction of millions of hydrogen bonds is quite strong. All right, again, our components of our DNA is going to be the base right here, that nitrogenous base, the sugar right here of deoxyribose, and the phosphate groups. And here you can see the base pairs forming the ladder. When they talk about the ladder structure, it's just the two bases, adenine and thymine, cytosine and guanine, going in the anti-parallel structure of DNA. And when they talk about anti-parallel, it's that all these phosphates and pentoses are pointing up on one side and then pointing kind of down on the other side. So they run anti-parallel to each other, like your hands going like that. Okay, so when DNA sitting in the nucleus of your cell needs to get its message out to start making some proteins, your DNA will be transcribed into messenger RNA specifically. So DNA is transcribed into RNA. That RNA will then travel out of the nucleus and go to the ribosomes, either the free ribosomes that are floating around in the cytosol or the ribosomes that are studding the endoplasmic reticulum, making it rough endoplasmic reticulum. Okay? And you will have enzymes on those ribosomes that will synthesize, well, first this is talking about the enzymes, that synthesize the complementary strand of RNA from the DNA template in the nucleus, okay? Once that's synthesized and RNA travels out, it travels out single-stranded, and RNA will then get transcribed on the ribosomes. RNA is different from DNA in that it is single-stranded and it uses ribose as the sugar instead of deoxyribose. And the third big difference is it uses uracil to replace thymine, okay? It uses uracil to replace thymine. So if you start seeing uracil, you should be dealing with RNA. It would be abnormal for it to be inserted in DNA, which happens occasionally, and that becomes a mutation. Okay, so our ribonucleic acid composed of the sugar ribose, it also has the phosphate group and a nitrogenous base. And some of our RNA molecules are temporary copies of the segments of the genes of DNA. That's our mRNA, if you want to put that as a notation there. And some of our RNA molecules will also be regulatory or act as enzymes. So you'll have some RNA-based enzymes on your ribosomes, and they're called ribozymes, okay? Forget about all that RNA interference. You don't need to know that now. That's from a different class. Okay, and then we have our transfer RNA that will bring our amino acids to our messenger RNA, where on those ribosomes, we're going to start hooking up our amino acids one to each other and making our peptide bond, right? Peptide bond. Okay, so we have an attachment site for our amino acid that'll be hooked on here. You have this clover leaf structure of transfer RNA that's going to come in, and you have an anticodon on your transfer RNA, and that 3 nitrogenous base sequence there of the anticodon on transfer RNA will match up to a codon or 3 base sequence on messenger RNA, and they match up exactly, and that's how you know which amino acid to bring over to the messenger RNA and start hooking everything together on our ribosome. All this should be a review for you, but I know it's fast review, okay? Is there anybody who didn't take Bio 155? It's supposed to be required. Okay. All right, so our nucleotides will have other important roles in the body, and the big one that we have is ATP. So our ATP also has nucleotides in it attached to high energy phosphates, okay? So it's adenosine triphosphate. And ATP, you will always know, is the energy currency of our cells. This is how we move energy around to drive all the reactions of metabolism in our body. All right, and when we do that, we use ATP. We typically split off a phosphate group, and ATP becomes ADP, and that inorganic phosphate is usually driving some of the enzymes to make some of these reactions. We have a couple of other high energy compounds, and these will be our coenzymes when we talk about our tricarboxylic acid cycle or our Krebs cycle. The thing that you never ever wanted to look at again after you got out of Bio 155. And I said, it will come back to haunt you if you didn't learn it. Well, here it is starting to come back to haunt you, okay? So NAD is nicotinamide adenine dinucleotide, and you can see why we just always refer to it as NAD, and FAD, and these are the coenzymes that get reduced, or they pick up hydrogen in glycolysis, okay? And eventually those reduced forms, that extra hydrogen bond in there, or bond to a hydrogen being reduced form, will have extra electrons, which then will split off in the electron transport chain so that we can make more ATP, okay? All right, so our ATP looks like this. We have our adenosine group, our nitrogenous base. We have a ribose sugar, just like in RNA and DNA. And then you have these 3 phosphates at the end at ATP. And if you split off one, you have ADP. If you split off another one, you're down to AMP. So adenosine triphosphate, monophosphate, and diphosphate, okay? And we split off the phosphates and ATP to get the energy to drive our reactions in our bodies. NAD also looks very similar in that you have your base up here, you have some phosphates in it, you have ribose sugars, and you have this other adenine base over here, okay? And like I said, we're going to build high energy molecules by taking our NAD+, which is the oxidized form, and we're going to add electrons and hydrogens to it and reduce it. This is called the reduced form, okay? It always seemed weird to me that we call it reduction when we're adding hydrogens because we're adding things to it. But, you know, hey, I'm not the chemist who named these things, all right? It was left up to men. Ha, they're so disorganized. No. I'm not a man hater, truly. I'm not. I just like to make fun of them. I love the men. All right, so NADH is our reduced form, and that's the high energy compound. And we will take this high energy molecule later on, and we will shuttle this into the electron transport chain in the mitochondria and take these hydrogens, this proton, back off of it and use that to form more ATP. Okay, so that's why it's cyclical. We're always recycling our coenzymes. Okay, so a lot of our molecules, we're going to have combined forms of these molecules, okay? And you've already seen this because sometimes we're going to have not just a lipid, but we'll have a phospholipid. And the names will tell you what's in them. So you'll have a lipid part of it with a phosphate group stuck onto it. ATP, adenosine triphosphate. The name tells you it has the adenine base and it has 3 phosphates. A glycoprotein has a protein, so you're going to have that peptide bond, a long chain of amino acids, but it's going to have glyco attached to it. Glyco meaning glucose or sugar, okay? So just be aware of that. I want to teach you the names so that you can just kind of figure it out as you're reading along. You know, fake it till you make it, right? All right, so this is, I think, our last slide. So as we get to all these biomolecules, a lot of what we're going to be concerned about, because we're ultimately getting as our goal to treat patients and their diseases. We don't actually in healthcare tend to see a whole lot of healthy people, especially now with COVID. God knows if you're healthy, you are avoiding healthcare like the plague because we have the plague. So we have disorders involving all these different types of macromolecules. Anybody know what particular disorder that's epidemic in our country that involves carbohydrates and our inability to process them well? particularly involving the carbohydrate known as glucose?
Diabetes.
Diabetes, exactly. So we have type 1 and type 2. And type 2 is the one that's plaguing all of us so much, because type 2 we get a little bit from what we do to ourselves. We have a genetic propensity for it, but then we eat too many donuts and Snicker bars and don't get enough exercise. And I'm guilty, right? Okay, I raise my hand right here. I'm guilty of this as everyone else. And I need to get a lot better about not consuming carbohydrates. But I mean, while you're all at home with this COVID plague being around us, sometimes don't you just want the comfort of a pizza and a beer?
Mm-hmm.
Yeah. All right. Or chocolate, whatever, pick your poison, right? But do realize it's poison. Okay. We'll also have disorders involving lipids. We already talked about hypercholesterolemia, nice long name for having high cholesterol, in which we give you medication like a statin to reduce your liver making too much cholesterol in your own body. We'll have disorders of protein function, and we'll have some disorders with nucleic acids that we'll talk about later on. These will be things like our genetic disorders, right? All right, so let's go through some practice questions, and then we'll take a well-earned break, but let's see if we learned anything that actually sticks in our brain. Okay, which of the following is not one of the major groups of organic substances in the human body?
Salts.
Yes, all right, good job, Early. So salts are inorganic. Proteins, lipids, and nucleic acids are obvious, are organic, and carbohydrates would be the other one. Okay, the amino group in an amino acid is?
E, symbolized by the letter R.
Nope, good guess. The letter R, the R group is what makes each particular amino acid unique. But all amino acids have a nitrogen group.
Nitrogen group.
Nitrogen
Okay.
group. Yeah, that's a lot harder question. Okay. Peptide bonds join together molecules of? So when we're forming our peptide, we knit together
Amino acids.
amino
Right.
acids. Good job,
Yeah.
Jasandra. Okay. Amino acids. All right. The element that's present in all proteins but not in carbohydrates is?
Nitrogen.
Nitrogen, right. And nitrogen is present in proteins and it's present in nucleic acids. Okay. Which level of protein structure refers to the number, the kind, and the sequence of amino acids? So just the sequence: valine, histidine, leucine, glycine, and so forth.
Primary.
Primary, great, good job. Jasandra, you're rocking out.
All right.
Which lipid acts as a tissue hormone? Remember I said, as soon as you see plasma membrane, you think phospholipid bilayer. As soon as you see this word, you think tissue hormone.
Prostaglandin.
Prostaglandin, good job. All right. Types of lipids found that form hormones such as cortisone, estrogen, and testosterone. What type of lipid is estrogen and testosterone?
Steroids.
Steroids.
Good job. Bria and somebody else I heard jump in too. Steroids. Okay, amino acids frequently become joined by what kind of reaction or bond?
Peptide bonds.
Peptide bond. Great. Hey, you guys are awake this morning. Phospholipids have a fat-soluble end and a water-soluble end.
True.
True. That's our membrane, okay? Most of the fat found in the human body is in the form of?
Triglycerides.
Triglycerides. Yep, think of that on your thigh, right? Your thigh and your butt and your belly. That's where we get it all, right? Thigh, butt, and belly.
Mhm.
The nitrogenous base is not found within the structure of DNA,
Uracil.
Uracil. Man, you were quick on that one. Okay. Which of the following is needed to form a triglyceride? Three of what? Three of what?
Fatty acids.
Three
Fatty
fatty
acids.
acids attached to one
Glycerol.
glycerol. So,
Glycerol?
yeah, there you go.
Oh, okay, E, okay.
Okay, whoops, we had it. I screwed up, so now. All right, well, food contains organic molecules of carbon, hydrogen, and oxygen in the ratio of 1:2 to 1. So what class of compounds does this belong to? And what's the function of that compound?
The carbohydrates and the energy.
Carbohydrates, 1:2 to 1. So C6H12O6 is typical sugar, right? Like glucose. And so it supplies energy is our carbohydrates. Lipids are low in oxygen. Proteins, you would see nitrogen. Nucleic acids, you'd also have to see nitrogen. Unsaturated fats. What's unique about unsaturated fats?
Where's your cup?
Saturated fat.
Do unsaturated fats have double bonds or single bonds?
We have double.
Double.
Okay,
It will be D.
so it will be D. Yep, they're kinking because of,
Okay, the kinky, okay.
all right. I almost want to start singing a Rick James, like it's a very kinky fat instead. And if I start singing, you guys are going to start running away. But you know, sometimes you make up the songs. This is another clue for anatomy, is you make up the songs, little songs, and the dirtier, filthier, the better. When you start like making little mnemonic devices for remembering your bones and muscles, you can look them up online. But you know, as I am your instructor, I should not be telling them to you myself. But they actually do help you remember things because you feel a little, you know, little jiggy in your brain there, okay? All right, so which of the following is not true of carbohydrates? Are they called sugars?
Oh,
Carbohydrates? Yeah, they're sugars,
Yeah.
commonly called sugars. Are they the primary source of our energy?
Yes.
Yes.
Yeah. And do we have sugars in DNA and RNA? Ribose and
Yes.
deoxyribose, so everything.
Mhm.
Everything, okay.
All right, cool. We'll take a break and come back, or you want to just keep going?
Take a break.
Take a break. That is a lot.
Listen.
Okay. Do you want me to sign off, and that way we can break down the recordings into smaller things, and I'll send a new link to everybody via email? Okay, I'll do that.
Okay.
I have a question.
Yep.
When are you gonna be sending the recordings on Blackboard? Do you send it like after every class? or-
I post it. So I've posted them on Blackboard, and I've posted them on DropSend. Here, let me show you where it's posted right now on Blackboard while we're here.
Okay. I'm glad she asked that because
Yeah.
I had the same question. Last time I had an issue with getting in with the recording. So I don't know if it was me or, you know.
Okay. All right. Let me see. I got all these different Blackboard accounts, so I got to get to WCCD. Right. Let me get back to screen sharing so you guys can see it, what I'm doing here. Okay, so my Blackboard's going to look a little bit different compared to your Blackboard. And so you get into the class, because I have multiple classes. This is the home page. I set it up as modules. So if you go over, oh, and I can kind of do it like what yours looks like here. I'll go to a student view. Okay, so student view, if you go over to modules, you can look here, and I've only put up all of part 1 right now, body organization. So you click on that, and you can see I have all this stuff here. So there's a section that's Chemistry of Life video series. And are your Chemistry of Life videos there? So you can click on one of those, and you can download it, Okay, Chemistry of Life, part 1, 2, 3. So those goes through all your chemistry things. You can click over onto the lab and you'll see for the lab, I have all this, organized as your PowerPoint that we went over the lab, the anatomical terminology for the lab, the body organization with your assignment and its due date.
Okay.
You can click on, oops, cancel. You can click on, I'm not used to navigating around in the student view. You can click on the lecture materials. Maybe here, let me exit the student view because I'm going to screw something up. Okay, if I go back to the modules, maybe, and one of you, like Charity, I know you're probably our real good person at navigating all this. When I organized it, I put everything in folders so that when you go into the body organization folder, you can see all the chemistry videos at once, all the lab 1, all the week 1 lecture materials, going down, all the week 2 lecture materials, all the week 3 lecture materials, all the membrane transport for the lab that we're going to do this week. And then hopefully in the next couple of days, I'm going to put your first problem-based assignment. So like when you go in to do lab 3, I have it set up like this is going to be the presentation we go through. This is sort of stuff about the experiment we're going to do today. And the one that's actually your assignment that you have to turn in has this little symbol on it of an assignment. So when you go into it, it shows you how many points are possible and you can do it on your computer. And this would be where I'd have to have someone like Charity or one of you can sign in and show everybody that you click on the assignment and you can do it here and you can load it back up in here and it just comes to me through Blackboard. All right.
Okay.
Wait, you said we can do it on the computer?
Yeah, you can do it on the computer. I made it as a fill-outable form. I'm trying to make it as easy as possible for you to do.
Okay, yeah, because I was gonna ask that. That's my second question. Because I downloaded, I clicked on the file and I was able to write it down on the PDF form. Like it came up on there.
Mm-hmm.
But I didn't know if there was also another way to do it, if that's the easiest way.
Yeah, that's the easiest way. I want to make it easy both for you guys to do it and turn it in and for me to grade it. Last semester when we got everything thrown in, I had people photographing things on like note paper sheets sometimes, and it makes it, when I get it 25 different ways, it makes it really hard to grade that way.
Yeah, I think it's really easier this way because it's just, you can just write it down. I used the Acrobat and it just worked out fine.
Yeah, and it just fills it out. So I'm trying to stick with that. Learning the anatomy is hard enough. I don't want you guys to have it even made harder by having all kinds of computer weird glitches. So I also put on this left side here, you have your home page. So if we have an announcement, see, here's all of you, right, thinking of coronavirus in the background. I have your syllabus and schedule here. I don't know if you all looked at the new syllabus and schedule, which I also just changed again and tweaked because instead of doing skin and epithelial lab, I wrote in that we're going to be doing this membrane transport lab instead. So, and that's fine. You can keep, it's just, the lab content is different. Everything else is the same. So you can download that and look at that as you want.
Yeah. I like how you color coded that, and I appreciate that, the way you color coded things to, to differentiate the two.
Yeah, yeah.
Thank you.
You're welcome. Thank you for appreciating that. And then I also have over here the links to my YouTube channel. So if you wanted to go to YouTube and watch some of the videos, you have a direct link here, and you can see I have things like the chemistry of life. This is part 3.
Hi, everyone.
And it goes in and we'll go through, all the stuff about all the videos. You can download this on Blackboard if you wanted to, or you could link to YouTube, or you can download it on DropSend. Okay. So I have lots of videos that I've been putting up. And then when you turn things in, it'll come over into the grade center, and I'll get the grading, and you'll see you all pop in here, and I'll get out of that fast so nobody sees anybody else's secret numbers. But I I don't really have a lot of grades recorded yet. I have to start sitting down and grading all of Lab 1. I was kind of waiting for a whole group of you to turn it in before I grade it, because it's easier for me to grade 10 in a row than it is to grade 1 on Monday, another 2 on Wednesday. But I should get those done this weekend sometime.
Ms. Harold, are we gonna for our textbooks? I know the textbooks are really helpful because I have mine open right here.
Mhm.
It helps with the homework.
Yeah.
You also said we needed an atlas.
You don't need an atlas, but
okay.
you might want an atlas. A
Yeah,
lot of students, I always found in labs, I usually bring like 3 of my atlases to lab, and students really like to use them. But obviously, you know, I can't do that for you now. So you might want to get one. And I would suggest you don't need, like, if you see Netter is what we use in medical school, and it's so dense, you don't need
I got
something.
the photographic atlas by Herbert Heisler.
Okay, yeah, look at something like that, and you know, you can go probably check one out at the library if you don't have one, and you know, even just your local library usually has anatomy atlases, and it might just help you look at things.
Yeah, I got one on Amazon for pretty cheap.
Yeah, so you can pick a lot of them up pretty cheap. Do we need to go over drop send a little bit? Is anybody still having trouble with drop send?
Do we actually need to use that, or is it fine if we just turn it in through Blackboard?
You can just use Blackboard.
Okay,
You can
because
just
Dropsend was extremely confusing to me.
Yeah, everything is on Blackboard, but
Okay, so
I also duplicated on drop send because sometimes some students I've found in the past have real difficulty. This class maybe not so much, but I've had classes where I have a lot of like 40-year-old ladies who are coming back to do nursing after, and I shouldn't say this because, you know, I'm aged myself. I'm 55 years old, but a lot of ladies who come back after raising their kids and they've been out of the workplace and they're not so good with their computers, and Blackboard is very overwhelming to them. And they've typically found drop send easier because they can just download the things on their phone, and they'll, you know, they're moms and stuff, and so they're busy and they're always looking on their phones. They're like studying while they're sitting out in their cars waiting for their kids to get out of school and pick them up and things like that. So I try to be adaptive for the reality of the life of what my students are. But yes, you can just use completely everything will be on Blackboard. You can ignore drop send if you want to, but it's there for you if you need it. Okay. All right. So time for break?
Yes.
Time for a break, and then I'll send you a new link, okay?
Okay.
It'll come in your email. All right.
Okay. Thank you.
Uh-huh.